Now we're going to discuss the concept of linkage and that of genetic mapping. First, we'll discuss what genetic mapping is, linkage, and how linkage is used in mapping, and then various techniques that geneticists use to figure out where genes are in genomes and on chromosomes. So, in general, genetic mapping is figuring out where genes are in genomes or on chromosomes. Physical mapping is determining the location of a gene on a eukaryotic chromosome, and we'll talk about many of the techniques that are used to do that. Linkage mapping is using the recombination frequency, how often recombination breaks up two genes that are linked or very close together on a chromosome. And then restriction mapping, which we won't really talk about much, is using restriction enzymes to cut DNA molecules and then figuring out where the enzymes cut in relation to each other. So the concept of linkage is the tendency for genes located close together on the same chromosome to be inherited together. And genes carried on the same chromosome are said to be linked. Linkage is the opposite or the converse of independent assortment. So linked genes don't exhibit independent assortment. In other words, they're not inherited independently, but they're normally inherited together, unless on the rare occasions when they are split up by a chiasma. So this graph shows that the percent recombination underestimates the physical distance at higher map distances. So remember that Mendel's theory of independent assortment states that each gene pair tends to assort into gametes independently of other gene pairs located on non-homologous chromosomes. And of course, this gives us that 9 to 3 to 3 to 1 phenotypic ratio in the F2 generation. So the probability then that crossing over will separate two genes on a chromosome is of course proportional to the distance between them. So it's crossing over that causes independent assortment. When two genes are located very close together, it's unlikely that a crossover will occur between them. Okay? And of course, the farther they are apart, the more likely they are to exhibit independent assortment. So more than 50% crossing over is considered independent assortment or non-linkage genes. Now with dihybrid crosses with no linkage, we see a 9:3:1 ratio for a typical dihybrid cross with complete dominance, 1:1 to 1:1 ratio with a dihybrid test cross. With linkage, there's no discernible ratios. We have two classes of progeny that are in large numbers. These are the parentals or the non-recombinants. They result from no crossing over during meiosis in the parent, but have the same genotype as the parents. Hence the term parentals. And the other class of progeny result from crossing over. So these are going to be in small numbers because we're dealing with genes close together. And so they're the result of crossing over. So linkage mapping then is using the amount of crossing over to determine the relative distance between genes. And also if we have 3 or more genes, we can figure out the order of the genes along the chromosomes. It's not absolute distance, but relative distance. And the distance we use is map units, which is percent crossing over, recombination frequency, or it's often called centimorgans, named after Thomas Morgan, a guy who was an early genetic pioneer and worked a lot with fruit flies about 100 years ago. So 1 map unit equals 1 centimorgan or 1% recombination. These are the units we use in linkage mapping. And here's the first guy to start linkage mapping, Alfred Sturtevant. He was a student of Morgan. And that's probably why he coined the term centimorgan. But anyway, you can see there he's got all his fruit fly vials on his table and lots of fruit fly pinups on the wall. And so his first map is shown here based on recombination. It's the part of the X chromosome of fruit flies, Drosophila melanogaster. And his symbols are above the line, B, C, P, R, and M, and today's modern symbols are below the line. So at 10 map units, we have the wide-eyed gene at 30.7 map units per million eyes, 33.7 miniature wings, 57.6 rudimentary wings. So I guess technically rudimentary wings wouldn't be linked to yellow body, for instance. They're more than 50 map units apart. Some of the various methods that scientists use, other than linkage, to determine the position of genes and genomes and on chromosomes include deletion mapping, somatic cell hybridization, C2 hybridization, and then DNA sequencing. And we'll discuss each of these in that order. Excuse me. So here's a map of the Drosophila genome, Drosophila melanogaster. The flies are 2n equals 8. So they have a haploid set of 4 chromosomes. Number 1 is the X chromosome, 2 and 3 are large chromosomes, and 4 is a very small chromosome with a few genes mapped to one end of it. You can see a lot of these different genes that were used a lot by geneticists. Here's a human chromosome map of chromosome 1, our biggest chromosome, and the X chromosome. And again, remember that the long arm is the Q arm and the short arm is the P arm. So you can see at towards the end of the Q arm and the X chromosome, we have CB for color blindness, next to that hema for hemophilia, for instance. So in deletion mapping, we grow cells in cultures, and we have some strains with deletions on the chromosome, like you can see there in the parental generation. And then we can then look at what genes are on what chromosomes. You can also get flies and various other organisms with deletions and do crosses with them to figure out where genes are on a chromosome. So here in this chromosome, number 2, I think of humans, the A breakpoint retained the gene ACP1, but the B breakpoint lost it. So that tells us that that ACP1 gene or locus has to be between A and B up here on towards the end of the P arm on this chromosome. So it's a little bit like a process of elimination, I guess you could say. And this shows you in 3 cell lines. Cell line 1 has human chromosomes 2, 4, 11. Cell line 2, 2 and 11. And then cell line 3, 2, 4, and 11. But in cell line 3, chromosome 4 has a deletion on the short arm. And so we can see that the protein product of the gene that we're interested in is present in cell line 1. It's absent in cell line 2 because there's no chromosome 4. And it's absent in cell line 3 because chromosome 4 has a deletion in the short arm. So that tells us then that the gene of interest is in that deleted region of chromosome 4, the little, as shown by the red bands there in cell line 1. Now, somatic cell hybridization. Here we take rodent and human cells, usually hamster and mouse cells, and we fuse those cells using viruses or chemicals, and that results in a cell with 2 cell nuclei called a heterokaryon. It's got 1 nucleus from each of the original cells. Then we can get the nuclei to fuse, and through successive generations, certain human chromosomes are lost, and we end up with a stable cell line. It's got all the rodent chromosomes and some subset of human chromosomes. Different cell lines have different subsets of our chromosomes. So, and then of course we can identify which chromosomes are present by their G banding patterns. With these somatic cell hybrids, we can do assignment tests, and that lets us figure out what gene is on what chromosome. For instance, the gene for UMP kinase was found to be on chromosome 1. We can also do tests for linkage called synteny tests, and they were able to figure out that the genes LDHB and peptidase B were linked together in the same linkage group with these kinds of tests. So this shows the process. So we have human and mouse cells, and they're put in a petri dish in the presence of polyethylene glycol, which facilitates fusion of the cell membranes. This creates those heterokaryons, a cell with 2 nuclei, and then eventually the nuclei fuse, and different human chromosomes are lost in these different cell lines. So here we're showing you 5 cell lines coming out of this experiment. So there's a standard karyotype. The chromosomes stain darkly, and you really couldn't pair them up very well this way. But remember, with G banding, you can, where each chromosome has a unique banding pattern, and so you can pair up the homologs and identify individual chromosomes that way. So this shows you how an assignment test would work. We've got 6 cell lines, A, B, C, D, E, and F. So what we want to do is figure out where on what chromosome a certain gene is located. The gene product, i.e. the protein, is present in cell lines A and B. It's present in cell lines D and F. And it's absent in cell lines C and E. So we merely look and see which chromosome is present every time the protein product is present. And we can see there that in chromosome 4, is present in cell lines A, B, D, and F every time the protein product of that gene is present. So for instance, in cell line B, it's got a lot of human chromosomes left, but not all of them. It's got human chromosomes 1, 2, 4, and 8 through 13 in it, okay? Whereas cell line D has only chromosomes, cell line C has only chromosomes 15, 17, and 19, and 22 in it. But anyway, we find with this that the gene of interest is located on chromosome 4 because every time the protein product is present, chromosome 4 is present. Okay, this shows how we use somatic cell hybridization to find some more examples of finding genes on chromosomes. So number 1, you make the fused cells. In 2, the hybrid cells are grown on hat medium to select for the TK plus and HPRT plus genes. During this time, human chromosomes are randomly lost from the hybrid cell lines. In 3A on the left there, selected cells carry the mouse HPRT plus gene and the human TK plus gene. Because human chromosome 17 is common to all selected cell lines, then the TK plus gene must be on human chromosome 17. Same line of reasoning over on the right there. Here we found that the HPRT plus gene is found in the X chromosome using this method. Now in C2 hybridization, here we take and hybridize a labeled probe to chromosomes that are already on a glass microscope slide. So in the context of molecular biology, hybridization means the annealing of DNA sequences from different sources. And then we take a probe, which is single-stranded DNA with the sequence of the desired gene. So that will, of course, anneal to the single-stranded DNA in the chromosomes on the microscope slide. And then in order to see this, we have to label the probe somehow. So the probe is either labeled with a radioactive isotope or else fluorescent dye so that we can see where on the chromosome it is hybridizing. And so FISH then is fluorescence in C2 hybridization. So we take the DNA in the chromosome and make it single-strand, usually treating it with something like sodium hydroxide. And then we take the probe DNA, make it single-stranded, and wash it over the microscope slide, and it should anneal to the single-stranded DNA that's got the complementary sequence. And this kind of shows you the steps involved there. At the top here, we squash the cells on a slide, treat it with 0.07 normal sodium hydroxide for a couple of minutes to make the DNA single-stranded, then incubate in radioactive DNA to remove the unhybridized DNA single strands, and then we can take and expose those radioactive cells to X-ray film, where if there's radioactivity, that will show up as dark grains or dark spots on the X-ray film. Or we can use a fluorescent dye probe as shown at the bottom here, which is stuck to an avidin molecule, which is stuck to a biotin molecule, which is directly stuck to the hybridization probe. So the little DNA sequence there is the probe, and then the big thing to the right of that is the label. So here are human telomeres. Remember, these cap the ends of chromosomes, and here they're showing up as yellow. So notice the ends of every chromosome has yellow on it. Those are the telomeres. This shows you fluorescence in situ hybridization, where chromosome 9 stained with red and chromosome 22 stained with green. So you can see a green and right in the middle, and then the 22 and the 2 9s up at the top of this karyotype. There's also something called a spectral karyotype, where each different chromosome is stained a different color. And that's a little off topic, but it's so pretty and interesting I had to show you. And then lastly, DNA sequencing. So this is determining the nucleotide sequence of a desired segment of DNA or a gene. It yields the physical distance nucleotide between genes. That way we can say the two genes are 10,000 base pairs apart, or 400 base pairs apart, or whatever. Nowadays it's inexpensive and fully automated, and there's different ways of doing it. But anyway, the entire genomes of many organisms have now been sequenced. But again, that bacterium, Haemophilus influenzae, was the first to have its genome sequenced back in 1995. So from chapter 7, I want you to do problems 18, 22, and 37. And remember, they're due at the end of the week.